thank you everyone to invited me to present today. So it will be quite related from the previous talk because it's also about mixtures. But here we are talking about individual plant mixture and between species, and as Timothée said, varietal mixture from both species at the same time in the same plot, and try to disentangle all those interactions to show if we can estimate something. So as previously said, our study is about agroecology and more precisely about diversification of crops within field. And we are focusing here on the species level, so intercropping, but also on genotypic level, varietal mixture at the same time. And so in our study, we really want to understand a little bit more the genetics behind interaction and among and within plants. So our research question is how can we accurately estimate a large number of inter- and interspecific plant-plant interaction, genetic effect in a reasonably sized experiment? Because as previously shown, the combinatorial exposure is a major problem in that field. So our framework is well, mostly the same. The labels are not the same, but it represents the same thing. So here you have 3 plants. 2 are wheat or durum wheat, and 1 is an alfalfa. And we can decompose the phenotype of the focal plants by one genetic effect that is called the DGE. So it's the same thing as the DBV presented before. So it's direct genetic effect, and some indirect genetic effect that's called IgE. So here you have 2 wheat plants. So the IgE from this plant, so it can be another genotype or the same genotype, will affect the focal plant that is next to him. So we call this intraspecific IgE in the presentation. And after that, we will also have a genetic effect from the alfalfa genotypes that will affect also the focal plants. That can be the same species or other species. And here, if it's the different species, it will be called interspecific IgE. And also we have environmental effect that we'll not take into account today. So to estimate those effects, we conduct a container experiment. So here you have a picture of the design, and we have some containers which are roughly 1 meter square of real soil with alternate columns from durum wheat and alfalfa. So here you have a design scheme to explain it. Like B and A are both species, it can be reversed. And we have a distance between plants in row and column that is the same, it's roughly 5 centimeters. And so to the genotypic aspect, we have 181 durum wheat lines from a diversity panel that is usually diverse. And in all the containers, it represents 1,920 plants that are randomized between containers and within containers. So you don't have any times the same neighborhood from every plant. Also, for the alfalfa, we have 106 alfalfa families that represent also 1,920 plants that are also randomized. So, as previously, I showed you that there was only one IgE from intraspecific and interspecific, but in this case, it will have a lot more interaction. So we will have DGE, that is the effect of the focal plant genotype on itself, so classical way. The IgE interspecific will be 2, like we are working on the other one of neighborhood. And we will have 2 IgE, intraspecific IgE, that affect every focal plants. And also on the interspecific aspect, we will have 6 IgE that affect every plant from which species, but also alfalfa 1. For the traits, we decided to measure 4 traits. So for the wheat, we have the plant height, heading time, upper leaf nitrogen content, and tiller number. From the literature, we know that height is directly impacted by light competition, and upper leaf nitrogen content or flag leaf nitrogen content and tiller number are highly dependent of mineral resources and mostly about nitrogen. And on the counterpart, we know that heading time is a very constructive conservative trait with no or minimal interaction between or G by E. For the alfalfa, we measure dry biomass and shape or growth habits. So both of those traits are very integrative, and so they respond to a lot of environmental conditions. So we didn't test it out. So here is the big one, the model. So at the phenotype, the model is a multivariate estimation model, and we use the model to estimate both species of genetic effect at the same time, because one is affecting another, and we have to make it together. So at the P, we have the vector or all phenotype from both species, A and B, in a long format. And after that, we will make some estimation. So the first line in this model is estimated thanks to the phenotype of species A and not phenotype from the species B. And the second line is genetic effect and means estimated with phenotype of species B and not phenotype of species A. So it's very structured. After that, we have on the left, on the right for you, the mean phenotypic value. After that, we will have the DGEs, so the direct genetic effect. So mostly the same thing we are measuring when we are talking about breeding value. We have the intraspecific IgE. So here is the genetic effect from wheat on wheat and from alfalfa on alfalfa. And after that, we have interspecific IgEs that are effect from one species to another. Thanks to this model, we also measured the covariances between those effects at the same time. So, for example, for the wheat, we have the GGE, interspecific IgE, and the intraspecific IgE that are simulated jointly in the same model, and we can estimate the variance-covariance matrix between those effects, genetic effect, to know and detect if there is patterns in the competition or that kind of thing. And we did the same for the other species. So here's the result, some of the results. On the bar plot, you can have you can see the partition of the phenotypic variances. For the heading date, in yellow you will have the GGE variance. On the blue you will have the intraspecific IgE variance, and in red you will have the interspecific IgE variance. So it's here the trait are for the wheat. So as a phenotypic variance, it's the IgE, interspecific IgE from the alfalfa that is affecting the variance of the wheat. So the red bar is not a genetic effect of wheat, it's a genetic effect of alfalfa. So thanks to that, we can already say that we have some variance, genetic variance that is due to alfalfa genotypes, and we can read it directly on the wheat phenotype. So if we only had wheat phenotypes, individual phenotypes, we could also conduct selections of, on alfalfa, even if it didn't measure any phenotypes of alfalfa. And to discriminate different traits, as I mentioned before, we are mostly computing the ratio between variance from the sum of the variance of the IgEs compared to the variance of a GGEs, that is the classical way. And so for the heading date, for example, we have 8% ratio that is corresponding to a low IgE over IgE traits. And it's directly linked to our assumption that heading dates is not really highly interacting with its environment. We have some medium IgE over the IgE trait, so if the plant 8, it's a 19% ratio. And after that, we have some traits that have a lot of IgE variance. So it's a lot of variance that is mostly not used in breeding scheme today compared to the GGE variance. So it's for tiller number and flag leaf nitrogen content, which represents 64% and 86% ratio. So we found large significant effect of IgE on several important developmental traits that can be used for breeding if you want to. And after that, as I explained it, we also have the covariances between those effects. So here is a representation of the G matrix. And here in yellow, you still have the GGE. In blue, you still have the intraspecific IgE. But here in red, it's the effect of the wheat on the alfalfa, not the effect of the alfalfa on the wheat. And so here you can see that there is a point that is surrounded that corresponds to a negative correlation. For example, it's just an example about when the wheat, leaf, nitrogen, GGE increase, there is a signal on the neighbor alfalfa biomass. So for me, it's a clear signal that it's probably competition for resources. And so when you have more resources, more nitrogen in your leaves, you'll probably take it from the soil and compete with alfalfa that reduce their biomass. And so thanks to this kind of G matrix, you can easily see that for breeding application, it would be very interesting to know how our indirect breeding effect will occur when you try to improve wheat nitrogen, for example. Also, this effect can be used to estimate or find association. And so here you have an example on wheat IgE until a number where we found a very strong QTL strongly associated with intraspecific IgEs. And we also found some on interspecific IgEs of alfalfa on wheat and wheat on alfalfa. So to conclude, this model allows us to jointly estimate a large number of interaction genetic effects across multiple species at the same time on a platform that is roughly the size of this area. The analysis of covariance structure help reveal how interaction are shaped within and between species in the same environment. And also, we can probably found and identify and average future breeding QTLs that can be used for intercropping and varietal mixture. I want to thank all the people involved. It was a lot of work because we have to phenotype a lot of plants. And thank you for your attention.